Okay, so this is no shade at all to this creator. It's just easier to reply to this question in video format. Whoa. So, gram-negative and gram-positive aren't like a infectious thing, and nothing's really causing gram-negative and gram-positive. It's just a structural difference, if that makes sense. So there's two types of bacteria widely, the gram-positive and the gram-negative. They're differentiated by the structure and composition of their cell wall around the bacteria. So this is the vague structure of a bacteria. It's no really specific one. It's just a bacteria. So this red around here is the cell wall. That's what gives the bacteria its structure. It does many other things, but that's kind of what we're talking about right now. This is two different gram stains. So this side over here, the purple one, is the gram-positive bacteria, and this over here is the gram-negative bacteria. I don't know specifically what types of bacteria they are. I just searched up gram-positive versus gram-negative. So the differences between gram-positive and gram-negative bacteria is the structure of the cell wall. Gram-positive bacteria have very thick cell walls with a bunch of peptidoglycan, which is the structural protein. Gram-positive bacteria don't have an outer membrane. They have the cell junk inside. They have the inner membrane, and then they have a very, very thick cell wall, which is what differentiates them from gram-negative bacteria. Gram-negative bacteria over here, they have way, way less peptidoglycan, the, again, structural protein, than the gram-positive. So the gram-negative bacteria have the cell junk, the inner membrane, a thin layer of peptidoglycan, and then a outer membrane, which is not made of peptidoglycan. Sorry, it's kind of late right now. I had a long drive today, so my speaking is not great. Now, gram staining is an identification test thing that you use to differentiate between gram-positive and gram-negative bacteria. That is important for identifying what type of bacteria it is. This is kind of the very first step you take in identifying a bacteria culture that you've grown, or that you just have. This is the first step in identifying the type of bacteria, which is very important for diagnosis and treatment if you have a bacterial infection. So the gram staining has a bunch of steps that I'm not really going to go into, but basically the gram-positive bacteria use the gram stain, which is purple, and it has the very, very thick peptidoglycan, which holds on to the purple color very, very well. Now, the gram-negative over here does not have as much peptidoglycan. It has a much thinner cell wall, so it doesn't hold on to this purple color nearly as well. Instead, you use what's called a counter stain, which is, in the case of the gram stain, bright red, to basically show that it's not this gram-positive color. And you need the counter stain in order to visualize the bacteria, because if there was no stain whatsoever, you couldn't see anything. So you have to have this red color to be able to see the bacteria. And now this is differentiated as gram-negative bacteria because it tests negative in the gram stain because it doesn't show up purple. So other comments by this person asked what does it mean when you test gram-positive? So you as an individual with a bacterial infection, you do not test gram-positive or negative because you're not a bacteria. Instead, the culture of the bacteria that was isolated from you, if you came in to get tested because you have an illness and you don't want to know what it is, there's usually a swab or a sample or culture, something taken from you. The bacteria are cultured, and the very first step is the gram stain to differentiate between gram-positive, gram-negative, to knock out a whole bunch of bacteria, because if it shows up purple, you know it's a couple certain types of bacteria, which you can do more tests for and you can find a treatment for. Same for if it's gram-negative, you know it's this specific type, and you can do more tests. Sorry, I'm really tired right now. You can do more tests to be able to determine what exact bacteria it is, again, for treatment. So nothing really causes gram-positive or negative bacteria other than evolution. It's just two different branches of bacteria that's separated somewhere. Some have a thicker cell wall and show up purple. Some have a thinner one with an outer membrane that show up red. Hope that makes sense. My brain is not working very well right now, but hopefully that clears some things up.